Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. I hope it wasn't too much of an inconvenience by doing a quick detour talking about urine alkalinization and urine pH and all that stuff to take a quick detour from our talk about glucose, but I felt compelled to do it, and I hope it was useful for you. So, getting back on track, talking about ways to manipulate the upregulation and reliance on glucose transporters, glucose related enzymes, part of the glycolysis system, so that the cancer cells can ferment glucose to make lactate. We focused last time on kind of discussing, really, are these of any clinical importance whatsoever? What are the subtypes of GLUT transporters, and kind of what research has found in terms of what transporters are normally seen on what subtypes of tissues. We know that the cancer cells require glucose as one of their Achilles heel. And obviously, our first line kind of treatment or intervention for that particular Achilles heel is going to be a therapeutic ketogenic diet and getting your blood glucose as low as possible, and then potentially shunting that glucose away from the tumor, whether we're using exercise or cold or something else. But moving forward to looking at ways to potentially inhibit and or downregulate the GLUT receptor, I'm going to go through a series of, I guess you'd call them, small molecule inhibitors that are designed or, being looked at by pharma. And then I'm going to look at the natural alternatives that we have. And one thing that I thought was somewhat interesting that we're going to talk about here is that there seems to be a synergistic effect between some of the natural inhibitors and the pharmacologic inhibitors. And I like that they're willing to go out there and say that there is benefit to these natural inhibitors when, being frank, they're going to be one to, patent and put their small molecule inhibitor to market. So they're being honest enough to say that there's still a role for these natural inhibitors. So without further ado, I'm going to start with this particular paper. And it was in January of 2020. It was published in Chem Biochem. And it says, and the title is Small Molecule Inhibition of Glucose Transporters GLUT 1 through 4. And it says, glucose addiction is observed in cancer and other diseases that are associated with hyperproliferation. The development of compounds that restrict glucose supply and decrease glycolysis has great potential for the development of new therapeutic approaches. Addressing facilitative glucose transporters, GLUTs, which are often upregulated in glucose-dependent cells, is therefore of particular interest. This article reviews a selection of potent isoform selective GLUT inhibitors and their biologic characterization. Potential therapeutic applications of GLUT inhibitors in oncology and other diseases that are linked to glucose addiction are discussed. And I pulled this article because it kind of precedes the next article, where we're really going to get into the actual paper itself. But I liked some of the graphics that it had. So for example, it had a great table here of all the compounds that are kind of being designed and pharmacologically studied, as well as their structure. And, it's going to say, it's a natural product, or it's going to be a small molecule inhibitor. And it's going to kind of use what marker they're using to detect kind of efficacy, and then what cell line they're actually doing, and then the actual concentrations in nanomoles that it takes to get it inhibited at 50%. And you can see here that some of these that we'll be talking about later on, like this Glutor, for example, is extremely potent at very low doses, whereas some of these other things, like this WZB117, takes about 100-fold more. So it's just it's pretty interesting to see these compounds be developed for the purpose of blocking these GLUT inhibitors. And then the next table, which is another reason why I pulled this paper, is for each of these compounds, it tells which GLUT transporter or a glucose transporter is actually being affected. So let's just use an example of cytokalasin B. It blocks GLUT 1 through 4, and then it's a non-competitive, whereas some of these are competitive inhibitors or mixed type inhibitors, and then it kind of gives like what some of their preclinical data at that time, and this was in 2020. So there's been updates to this. The next paper that followed that paper, it seems, is this paper here, and it's titled "An Appraisal of the Current Status of Inhibition of Glucose Transporters as an Emerging Anti-Neoplastic Approach, Promising Potential of New Pan-GLUT Inhibitors." And it was from 2022, November, in Frontiers in Pharmacology. And it says here that neoplastic cells displayed altered metabolism and accelerated glycolysis, or the metabolism of glucose. Therefore, these cells need a mammoth supply of glucose, for which they display an upregulated expression of various glucose transporters, the GLUT or GLUTs. Thus, novel anti-neoplastic strategies focusing on inhibiting GLUT to intersect the glycolysis lifeline of cancer cells. This review focuses on current status of various GLUT inhibition scenarios. The GLUT inhibitors belong to both natural and synthetic small inhibitory molecules category. As neoplastic cells express multiple GLUT isoforms, it is necessary to use pan-GLUT inhibitors. Nevertheless, it is also necessary that such pan-GLUT inhibitors exert such action at a low concentration so that normal cells are left unharmed and minimal injury is caused to other organs and systems of the body. I'm going to take a quick digression here and say it's going to be less of a problem if the person is in therapeutic ketosis, because they're fat adapted and the healthy cells are using ketones and you're utilizing that effectively, whereas the cancer cells can't. But I'm going to go back here. Moreover, approaches are also emerging from combining GLUT inhibitors with other chemotherapeutic agents to potentiate the anti-neoplastic action. A new pan-GLUT inhibitor named Glutor, a piperazine 1 derivative, has shown potent anti-neoplastic action owing to its inhibitory action exerted at nanomolar concentrations. So what this graphic is actually showing us here is that the problem is the cancer cells are upregulating these GLUT transporters. And there are various isoforms, which means there are various subtypes of these GLUT transporters: GLUT 1, GLUT 2, GLUT 3, all the way up to like GLUT 12, I think. I may be mistaken. There may be a 13th. My point is that they're looking at a variety of natural products, as well as kind of very, very novel things, like looking at like DNAs and RNAs and mRNAs as a way to do this. And then there's the small molecule inhibitors, which are being developed by pharma to help try to block these as well. And we're going to start looking at the non-natural things first, and then we can go in deeper and look at the natural inhibitors as well. So again, here's some structures of some of the things we're going to be talking about in this particular paper. I was able to pull out a lot of good information here. So the first compound is this WZB117. And it says here that it is a polyphenol-derived small molecule inhibitor of GLUT1. It exerts anti-cancer or anti-neoplastic action with an IC50 at 10 micromolar concentration on a variety of different cells, accompanied by enhanced radiosensitivity. So basically, it makes these cancer cells more sensitive to radiation therapy as well. And it says, interestingly, this WZB117 is also reported to inhibit cancer stem cells. However, one of the disadvantages of WZB117 is its instability, owing to its ester bonds under aqueous physiologic conditions, potentially the most significant limiting factor in its use in cancer therapeutics. Moreover, it is described as only inhibiting GLUT1, which can be another limitation in its applications, because some of these cancer cells are going to be upregulating several different subtypes. And so by blocking just GLUT1, that's going to be a limitation of its effectiveness, essentially. That was this WZB117. The next one we're going to talk about is this DRB18. And it says, so far, DRB18 has been shown to exert cytostatic, means stopping growth, action on around 60 different cancer cell lines of diverse origins. The primary mechanism of the anti-neoplastic action of DRB18 has been shown via increased endoplasmic reticulum oxidative stress. The pan-GLUT inhibitory ability of DRB18 was verified through in silico-based studies. Moreover, DRB18 treatment caused remarkable inhibition of glucose-dependent metabolic pathways like glycolysis, TCA cycle, purine, pyrimidine, that's DNA and RNA synthesis. However, the GLUT inhibitory and anti-neoplastic action of DRB18 has not been reported on any hematologic malignancies. That would be like leukemias, myelodysplastic syndromes, etc. Later says safety issues on normal tissues and organs of the tumor-bearing host also remain largely unverified. Moreover, DRB18 is water insoluble, which could also be another limitation to its use in cancer strategies. So this blocks more than one GLUT inhibitor or GLUT transporter, which is good. But at the same time, there's some limitations. One of the limitations is that they haven't, and this just is going to take further study. It's going to, they need to do some further safety studies, essentially. And they say that it's not soluble in water, so obviously our blood plasma is water, 99% water. So when it's not soluble in water, you mean you're going to have some kind of a carrier, and it just kind of complicates things for it to be, maximally effective for us going forward. So there's still some work that needs to be done, it sounds like, on that one as well. There's two more, this BAY876 and Fasentin, and that would be here and here. Since they were small little things, I put them on the same slide here. So this BAY876 is a quinoline-derived name. BAY876 was initially identified for its highly selective GLUT1 inhibition potential, by which it exerts actions in the nanomolar range, but has limited ability to inhibit other GLUT isoforms like GLUT2, 3, and 4. Additional advantages is that it does have solubility. It is soluble in water, basically, in vitro. Adequate bioavailability upon. So basically, they're saying that if you administer it via oral route, instead of having to be injected via IV or into your muscle or subcutaneous tissue, they're saying it is fairly bioavailable, but it doesn't have great effects against these other inhibition, against GLUT2, 3, and 4, essentially, which are also found on cancer cells. It says, however, docking studies have been performed to binding to GLUT1, and its exclusive binding to only GLUT1 limits its therapeutic applications. Moreover, the safety have also needs further verifications. So a lot of these seem to be in pretty early stages of study, and maybe there's just not any backing or any money going towards these studies, because it sounds like some of the studies, like with the safety stuff, probably they just need to get it done, and that's just going to take some time to get those done. But they're saying it seems that what I'm seeing by reading these is that there seems to be, at least according to these authors, if it only blocks GLUT1, that is fairly limiting in terms of its effectiveness by being used as a single agent, if that makes sense. The next one, this Fasentin. Let's see here. I put Fasentin leads to glucose deprivation by its ability to bind to GLUT1 and GLUT4, as revealed by docking studies. However, despite the promising GLUT1 inhibitory potential alongside sensitization of cells to death stimuli, not much has been worked out concerning its anti-neoplastic potential, which means I don't know how it even has been studied on GLUT1 if it is not looking at it from a cancer standpoint and as a improving, apoptosis. But I'll digress. A recent study reported that Fasentin could inhibit the proliferation of endothelial cells and hence may have implications in anti-neoplastic therapeutic strategies. However, its effect on other healthy normal cells must be explored to predict safety issues. So again, this one seems like it's even less along in terms of the pipeline. As you see, I think it was so it seems like they've tested this WZB against several subtypes, but this one has shown it seems to be further along, as shown by the fact that they've looked at it against 60 different cancer subtypes having an effect. So you're going to see that these are, although they're all in this paper and being discussed, some of them are further along in terms of drug development compared to others. The next one is this STF31. That is this one here. So STF31. So another promising GLUT1 inhibitory agent with anti-neoplastic potential is this STF31. And it says it was initially shown, it was shown initially to manifest renal cell carcinoma specific cytotoxic activity. Additionally, STF31 has been shown to modulate hypoxia factor, or HIF, and target NAMPT to manifest anti-neoplastic action as well. And later it says, however, the utility of the same will depend on expression levels of GLUT1 and NAMPT, which may vary in a tumor-to-tumor manner. Again, going back to, I think it was this slide here, we see here that, GLUT1, although it is probably the most important, GLUT1 and probably GLUT3 are the most important. Some of these, because they only block GLUT1 in a meaningful way, and they don't have any effect or sometimes very little effect on GLUT2 and 3, they're saying that really limits it. And some of these cancers will manifest several of these GLUT transporters, which means that, for me, it means, number one, it's probably going to require combination approach therapy, number one, of just the glucose transporters. Then, in addition to that, you're probably going to need to be using multiple strategies of inhibiting just the entire sugar burning process. So, for example, ketogenic diet, things that lower blood glucose, exercise, things that steal glucose away from tumors, cold, exercise, etc. And then blocking maybe one or multiple of these glucose transporters using hopefully one agent, but maybe a couple different agents. And then as we go down further in glycolysis, potentially blocking some of the rate limiting steps like hexokinase or phosphofructokinase, or potentially further on down like lactate dehydrogenase, et cetera, as a way to not put all of our eggs in one basket. So if we put all our eggs in, let's say, I don't know, Glutor, which blocks, we're going to talk about it next actually, but let's say it blocks GLUT1 through 3, but we put all of our eggs in that basket. If any amount of GLUT4 or other like transporters are expressed, then you have bypassed that, right? So again, kind of how the way we, and even mainstream conventional medicine, treat tuberculosis or HIV, we use 3 or 4 drug what's the word I'm looking for? 3 or 4 drug cocktails as a way to minimize reliance on one particular mechanism that the virus and or bacteria in these cases are using, as a way to kind of make sure it is killed. So I think that is kind of how it's going to probably have to work with metabolic therapies as well. And I think this gets into, some like from a 20,000 foot view, like, is this something that should be pressed, or is this something that should be pulsed? And I think that, personally, I think there's some controversy with that. I think that the way that it is not recommended, that's not a true characterization. The way that it is proposed and kind of outlined in metabolic therapy, from at least the first paper that was published in 2017, where you have kind of like this intermittent dosing of glucose and glutamine inhibitors, that may be the case for some glucose inhibitors, but that may be not the case for some other glucose inhibitors, if that makes sense. So, for example, like let's say metformin, it's going to have multiple effects within the glucose pathway. And so that could be something we do every day, right? For people with diabetes, prediabetes, or even who use it for as a kind of anti-aging type stuff. We know that could be done every day. But other things like maybe DCA, dichloroacetate, or 2DG, or maybe some of these very strong gluten inhibitors, maybe they can't be used every day. that's going to be kind of have to be worked out over time as we get more studies, especially human trials with these kind of things. Okay, that was this one. And the reason I went back to that other slide again was because what did it say here? It says um, the utility of this particular compound, this STF31, will depend on the expression levels of GLUT1 or this NAMPT, which may vary from tumor to tumor. Moreover, it is also reported that if a cancer is expressing GLUT2, they can overcome the inhibitory effects of that particular thing that's only blocking GLUT1. That's why, like I said, you may be on either a pan-GLUT inhibitor or a combination of things that kind of cover your bases from that particular area. But I still think that's even, in my opinion, that's going to be short-sighted. You're probably going to need to be doing multiple levels of this at the same time to have the maximum effect and really take advantage of the backbone of, or the Achilles heel of cancer, which is glucose addiction. All right, so this is the kind of seems like the granddaddy of the small molecule drug inhibitors, and it's called Glutor. Sounds like a superhero, a pan-GLUT inhibitor. And it says recently a pan-GLUT inhibitor named Glutor has been demonstrated to exhibit a potent anti-neoplastic effect or action in the nanomolar concentration range against cancer cells of diverse origin. Glutor was identified to inhibit GLUT1, 2, 3 in HCT116 cells. The inhibition of the uptake of 2-deoxyglucose was determined to have an IC50 of 11 nanomolars in most sensitive cell lines, including urinary-derived cell lines, which are the most glucose addicted. Interestingly, Glutor was found to impart no cytotoxicity on normal, healthy, peripheral blood mononuclear cells and Institute for Medical Research 90 embryonic lung cells. So they're saying that it seems to be very safe. The safety studies have been done on the other normal cells, at least. Furthermore, the elegant study of this author also screened the anti-neoplastic action of Glutor on a battery of about 94 cancer cell lines of diverse origins, and it observed that the anti-neoplastic action was exerted with an IC50 of less than 100 nanomolars. Later, it says, interestingly, Glutor treatment minimized glucose deprivation in a dose-dependent manner, aka the more you give this medication, the more it's going to block the glucose. Hence, Glutor displays the potential for effectively inhibiting glucose uptake in solid tumors. The sensitivity or resistance of Glutor depends on the cancer cell's metabolic background. The Glutor resistant cell lines can be can stitch between glycolysis and oxphos phenotypes. Depending on the metabolic makeup of the target cells and selected dose, Glutor could usher a complete glucose deprivation condition, even leading to upregulation of GLUT1 and GLUT3 expression due to hypoglycemia. Moreover, it is also likely that by its ability to cause inhibition of GLUT1, 2, and 3, Glutor possesses an extraordinary capacity to overcome the rescue and compensatory mechanisms of highly adaptive neoplastic or cancer cells. So just stepping back for a second, because it blocks GLUT1, 2, and 3, some of the rescue mechanisms that these cancer cells have is like, okay, GLUT1 is blocked, I'm going to upregulate 2 and 3. And so because it blocks all 3, it kind of checkmates these cancer cells because it's able to overcome the rescue and compensatory mechanisms of these highly adaptive cancer cells. Later, it says it is also noteworthy that the study of this author also indicated that the neoplastic cells' cytostasis and cytotoxicity could be synergistically upregulated by using Glutor along with glutaminase inhibitor CB839, which I'm going to talk about again, which is this paper here, okay? Let's see here. Later, it says, thus, the results obtained based on the screening of Glutor for its anti-neoplastic action on 94 different cancer cell lines derived from highly malignant cancers revealed the extraordinarily ability of glutor to inhibit glucose uptake, glycolytic flux, and cell survival. However, despite the evaluation and certification of the antineoplastic agent or action of glutor on cancer cell lines of diverse origins, the same has not been evaluated in any in vivo tumor model, which will be necessary to assess the translational value of glutor's antineoplastic potential for therapeutic applications. So what are they saying here? They're saying that in a test tube on 94 different types of cancer, glutor shuts them down completely. Okay. However, and I'm guessing this is probably a funding issue, is that they have not been able to study this on any animal or human model yet, in vivo, essentially. Let's see here. Moreover, the issues of bioavailability and ideal administration routes also need to be assessed in an appropriate in vivo model. They have not got to the point where they're even knowing how well it absorbs because it's so early in study. Glutor treatment of tumor cells inhibited cell survival and increased cell death. It also caused a decrease in glucose uptake associated with altered expression of GLUT1, 3. So not only does it block GLUT1, 2, and 3, it actually inhibits the expression of GLUT1 and 3, probably, and hexokinase 2 lactate dehydrogenase, probably because it's having an effect on hypoxia-inducible factor, which is known to upregulate all of those things, including this. I think this is a lactate transporter as well. Also decreased with diminishing lactate production because glycolysis is being shut down through various mechanisms here. Obviously, the lactic acid production is going to collapse and be downregulated, and it also diminished the deregulation of pH homeostasis. So someone in the comments, I don't remember who it was, I'm sorry, had mentioned, I think he said, my gut tells me that although these gluten inhibitors could be beneficial, my gut tells me that by just being on a ketogenic diet, that's going to normalize the tumor microenvironment and diminish the lactate production. And the answer is absolutely it will. Seafried has basically said the same thing in a paper, but just by adding these GLUT inhibitors such as glutor, you're going to also, in addition to blocking it and causing cell death, you're also going to have a diminished lactate production, which is also going to help decouple or de-inhibit the immune system because the lactate tumor microenvironment is going to be diminished and the pH is going to be kind of restored to normal physiologic pH or closer to normal physiologic pH so that the immune system can actually turn on and work, which is pretty cool also. And then lastly, it says that chemosensitivity of tumor cells increased following exposure to glutor and decreased MDR expression. Hence, multiple drug resistance 1, I think is what they call that, expression. Hence, these observations indicate that glutor has the potential to be used in antineoplastic therapeutic applications. And I could not agree more. I think this seems to be a very promising small molecule that I think could dovetail beautifully into metabolic therapy as a whole. Maybe people will laugh who have worked with me when I say that I would, of course, like to use less, not more, because some of the protocols that I build are pretty extensive. But that being said, if I could have something that blocked, was a pan glucose inhibitor and then maybe a pan glutamine inhibitor, and you're only kind of taking 2 things instead of like 10 things, that would be pretty cool, in addition to your ketogenic diet. All right, moving on. So later in the paper, it does talk about some of the combination approaches, and I thought that was interesting for our study here. So combinatorial approaches. Studies have indicated that promising potential of combinational approaches for GLUT inhibition instead of a single inhibitor approach. Such approaches mainly involve a combination of agents with, number 1, the ability to directly bind to the GLUT isoform and cause their inhibition. 2, approaches that inhibit expression of GLUT, basically cut it off at the pass, before it's even, made by the DNA and nucleus to be made into a protein to be put onto the cell membrane. Number 2. Number 3, the use of agents which compete with glucose for binding of GLUTs and hence cause inhibited glucose transport. Further, to achieve success, such approaches, GLUT inhibitors and pan-GLUT inhibitory potential can be of great use to usher the inhibition of multiple GLUT isoforms. Moreover, a combination of multiple GLUT inhibitors of natural and synthetic origin can also be considered and has shown hope during in vitro experimentation on cancer cell lines of various origins. However, adding several inhibitors together may cause toxicity and lead to certain harmful effects on normal healthy cells. I do believe that probably is going to be mitigated to a large degree by being keto adapted, by being fatto adapted, by being on a ketogenic diet. I'll digress. Which also use GLUT isoforms for glucose uptake. Furthermore, most of such combinations still lack the much required in vivo testing for evaluation, efficacy, and safety parameters, which is a big deal. They need to really get on the ball, and that's going to require likely funding for that to happen, and hopefully the will for them to actually get those studies done, because I think it's going to benefit all of humanity by doing these studies. Further on, it says the most promising of such combinations with successful antineoplastic outcomes include the combination of GLUT inhibitors like cytochalasin B, WZB117, and its derivatives, BAY87876, silibinin or milk thistle, Glutor, STF31, and fluoretin, with other conventional anti-cancer drugs like doxorubicin and curcumin. I thought that was interesting. They call that a conventional anti-cancer drug, curcumin. But anyway, etoposide, cisplatin, 5-fluorouracil or 5-FU, then cysteine, cytarabine, oxaliplatin, paclitaxel, and antimycin A. So, and then later, which is what I'm going to highlight in this particular paper here. For example, the combination of Glutor and glutaminase inhibitor CB839 resulted in successful antineoplastic manifestations. Additionally, the combination of GLUT inhibitors with radiotherapeutic strategies has also been displayed improved antineoplastic potential. Moreover, GLUT inhibitors combined with other glycolytic agents also result in superior inhibition of a tumor cell metabolism compared to such actions when applied alone. Again, exactly what I'm saying. Let's look quickly back at glycolysis, okay? This is just one example, okay? So what we're looking at here, everybody, is we're looking at blocking these glucose transporters, okay? And we talked about various compounds that block a variety of these glucose compounds or these glucose transporters, okay? Remember, there's many isoforms, and some of these glucose transporter inhibiting compounds like Glutor block GLUT 1, 2, and 3. Apigenin actually is more impressive. We'll talk about it when we get to the natural inhibitors. But my point is that what this paper is essentially saying, and I agree fully, is that although we could have multiple compounds blocking glucose transporters, to rely on that as a unique strategy or like a standalone strategy is probably going to not yield the best results. For example, we could, like that one paper and study was talking about, was blocking glucose transporters and using compound CB839 to block glutaminase, similar to the enzyme that is blocked by DON, okay, or mebendazole, or curcumin and I think berberine also have effects here, and betulinic acid, et cetera. But what I think they're also saying is that in addition to blocking glutamine and just the glucose transporter, it'd be better to block like glucose transporter and then hexokinase, HK, and then maybe lactate dehydrogenase. And then maybe there are some of these lactate kind of like transporters that could be potentially inhibited, as well as blocking glutamine. And I think the same thing kind of when we talked about the glutamine series, micro series, we talked about maybe using something that blocks glutamine uptake, glutamine utilization, maybe at multiple steps. There's also a GLUT enzyme here that is blocked by EGCG and a couple other things that converts glutamate to alpha ketoglutarate. And then in addition to that, there's multiple glutamine transporters, one of which is an antiporter that actually gets rid of glutamine to sacrifice it for bringing in cysteine to make glutathione, the SLC7A11 or XC transporter. And so my point is, and what that paper also is basically saying also, is that it's probably better to have kind of a a team approach to this where you have maybe multiple inhibitors of this, a couple inhibitors of this, a couple of inhibitors of this, and then inhibitors of the glutamine system as well, as a way to fully lock down and really put the screws in to the Achilles heels of these cancer cells, which are dependent and are addicted to glucose and glutamine. And I think that's very wise counsel by this author right here when it says that. Now, the other, I'd actually already shown this paper in the previous video, but that being said, this is the paper that in the study that this particular, part of this paper was describing, was this paper here, where they were using a glucose transport inhibitor. They were using Glutor, and they were using CB839 as a way to have a synergistic effect at blocking cancer metabolism. And I think that is definitely the way that Dr. Seyfried had laid it out by blocking glucose and glutamine, right? And I think that is going to be the way that's going to be likely the most effective. And I think that probably when you're keto adapted, you're going to be able to have a higher or a more wide, what we call therapeutic window with some of these medications, because you are kind of used to your other cells, the normal cells, are not dependent on glucose. Let me give you a for instance. So if you're hypothetically a diabetic and your normal blood glucoses are in the 200 to 300s, okay? If I give you insulin and I put you at, let's say 80 or 90, which is still normal, you're going to feel the symptoms is if you had your glucose of 30 or 20, okay? You're going to feel hypoglycemic because you are relatively hypoglycemic and you are not fat adapted, okay? If, for example, you are not diabetic, but you normally have your glucose at in maybe the high 90s, low 100s, and I put you down to 50, you're going to feel hypoglycemia because it's a relative decrease in glucose. However, if you are keto adapted and you have a good glucose and ketone numbers with a good GKI, in my experience, I have people get their blood glucose taken all the time at like Quest or LabCorp or whatever, and they get their fasting blood glucose when I check a comprehensive metabolic panel or a basic metabolic panel, and they can have blood glucoses in the 50s and 60s. And I'll be like, hey, when you went to go to Quest the other day, like, did you feel hypoglycemic? And they're like, nope, I felt fine. I felt good, actually, because they're keto adapted, right? So my point is that it would make a lot more sense to me that you use a compound like this. If you were to use a compound like this, once we figure out more about it, about how to dose it, how to get it into the body, it would make more sense, even if it blocks glucose uptake ports or transporters amazingly well. It would make sense to give yourself a little bit more of a therapeutic window to be able to tolerate these medications better if you were keto adapted. So even if these medications are like a silver bullet on the glucose side of things for cancer, I still think it would make sense to implement some degree of a ketogenic diet, so that way that you don't have the potential for as many side effects when we understand more about them. Someone had asked here, any idea of these gluten inhibitors affect insulin levels? Probably not. Probably not. I don't know why they would, because it'd be different if you were blocking, like, let's say you were blocking glucose uptake for the entire body, and you basically you couldn't get the glucose inside of the cells, the normal cells that are relying on the glucose from a more holistic perspective. Then I could see maybe like insulin being upregulated or increased because it thinks that it needs more to get the glucose inside the cells, right? So maybe in that context, maybe it could. I don't know for a fact that it could. And that may be another reason why, we would pulse these, we kind of press down some of the, things, and we pulse other things. So it's hard to really know. We have to know by, studying them when they start to do in vivo studies and they start to look at people who actually get these compounds. And then hopefully they're checking things like fasting blood glucose and fasting insulin to see if there's any changes. I don't know the answer to that. All right, this is like another cool graphic of glutor. Basically, you're seeing here that the GLUT transporters. These are supposed to be representing glucose molecules, these hexagonal structures, and glutor is blocking the transporter here. And then it's also the decreased glucose is going to have an effect on HIF1, as well as decreased lactate, and an increased pH will also have an effect on HIF1, because HIF1 alpha, even though it is a hypoxia induced factor, that's why it's hypoxia inducible factor 1 alpha, it also has other things that can induce it and stabilize it, and that's called pseudohypoxia. And that would be things like lactic acid, or the difference in pH, even glutamine when it's in excess, the ammonia from glutamine, as well as buildup of some of the TCA intermediates like succinate and malate can also stabilize hypoxia factors. So my point is that it also starts to relieve some of these drivers, I guess you'd call them, of the vicious cycles. And one of the major vicious cycles is the HIF1 alpha, because when HIF1 alpha is destabilized and it gets to be destroyed by the proteasome, then it stops doing its effects inside the nucleus, and the effects are multiple. It will increase glucose transporters, increase all the machinery needed for glycolysis. It will upregulate lactate dehydrogenases and the lactate transporters, as well as all the glutamine machinery as well. So, if you really wanted, kind of the root of the Warburg metabolism, at least a big part of it is going to be HIF1 alpha. So any ways we can destabilize hypoxia factors, that's going to be a good strategy for us. And we're actually going to have, matter of fact, we will have an entire, at least one lecture, maybe several lectures on natural inhibitors of hypoxia-inducible factor 1 alpha and ways to destabilize hypoxia-inducible factor 1 alpha. All right, going down further. So I just wanted to kind of like make sure I covered everything, because there are a couple other medications, ritonavir, an antiviral medication, as well as this fluoretin that is not in this particular paper as being one of the compounds. We did see facetin, and we did talk about it even, but there are a couple other, I guess either drug or small molecule inhibitors that are having an effect on glucose transporters. I also wanted to highlight that mebendazole also will target the GLUT1 transporter. It may have effects on other transporters as well, but we know that it has effects on GLUT1 at least. I would also mention here that says, based on the importance of metabolic reprogramming for the development of cancer progression, inhibitors of cell metabolism have been studied as potential candidates for chemotherapy in oncology. Mebendazole, an anti-helminthic approved by FDA, has shown antitumoral activity against cancer cell lines. However, its potential in the modulation of tumor metabolism remains unclear. That is actually, when was this published? 2023. That's interesting, because I think Seyfried, I don't remember when, I think it was earlier than 2023. I could be mistaken. I'll have to look at the publication. Seyfried is actually the one who discovered that mebendazole actually has effects on the GLS enzyme, similar to that of Dawn. And in that paper, he talks about it having effects on GLUT transporters as well. I don't remember if he, in that paper, mentioned the subtype of glucose transporter that I have to look back at. But it seems that it's been kind of known to have anti-cancer effects for quite a while. Maybe they didn't understand exactly why as well, but it's being elicited over the last at least 2 to 3 years, probably. It also, in addition to having effects on glutamine, which we've talked about, as well as glucose transporters, it also modulates the actual mRNA and the expression of GLUT1 hexokinase, this GAPHD, as well as lactate dehydrogenase A. I have to look and see what GAPHD is. I think it may just be pyruvate dehydrogenase, but I'm not sure what GAPDH is. But anyway, my point is that it's seeming like as we get further down the rabbit hole with mebendazole, that it's going to be pretty important, I think, for the purposes of cancer. I think that we already understand its role for glutamine inhibition. And I think that, again, it's being used in combination with temozolomide for for glioblastoma. And I was even seeing some pretty bold language. And one of the papers that I read saying that they believe that mebendazole could even replace temozolomide as a standard of care chemotherapeutic for um, glioblastoma. So, anywhere that we have a compound that has effects on glucose and glutamine at the same time, I think that's going to be probably a winner. And I think mebendazole is one of those. I wanted to highlight that. The other interesting kind of like compounds that could be helpful for us are the SGLT2 inhibitors. And it says here that it stands for sodium glucose link transporters. And the SGLT2 inhibitors are oral medications that help lower blood glucose or blood sugar in individuals with type 2 diabetes mellitus by inhibiting glucose reabsorption within the kidneys, which cause the body to excrete more sugar in the urine. There's inherent problems with that, like increasing urinary tract infections and stuff like that. But currently, there are 4 SGLT2 inhibitors that are commercially available or accessible in the United States, including canagliflozin, danagliflozin, etugliflozin, and empagliflozin. And then later it says that these molecules block the uptake of glucose in cancer cells, which lead to the death of these cells. Demonstrated that demonstrated that the SGL2 inhibition of canagliflozin directly inhibited the proliferation of cells in a mice-bearing liver tumor model. This study found that canagliflozin, I'm sure I'm pronouncing that wrong, but that being said, reduced the growth of liver cancer cells by blocking their uptake of glucose. In another study by this author, conducted an extensive study using Medicare-linked data and demonstrated that the initiation of these SGL2 inhibitors appeared to enhance overall survival with patients with hepatocellular carcinoma or primary liver cancer who already had diabetes compared to those not using them. I thought that's very fascinating. That may be like independent of the A1C and glucose getting to the or glucose level, right? Correspondingly, with other experimental studies have hinted at possible advantage of SGLT2 inhibitors when treating liver cancer. Let's be real. It's probably going to have a pan cancer effect because all these cancer cells are glucose addicted. That's why the Warburg metabolism and upregulation of glucose transporters are kind of a hallmark of cancer. So anyway, so that's kind of the drugs, portion of this video. And I'm going to kind of quickly, in the last maybe 10 minutes or so, go over the natural inhibitors. And it's going to be a little bit more streamlined and succinct, because what I was dealing with, you guys, was basically that I get papers and graphics that are cool, and it really helps me start to go down rabbit holes because it gives me a whole list of things that I can study, right? But it doesn't really tell me. I mean, this one it does. It says GLUT1, but in other ones like this, where you have a bunch of different compounds, and then it's having little arrows to different parts of the glycolysis system, it's very non-specific. And so therefore I wanted to make kind of a master list. Again, more of them. Here's your glucose transporter. Here's a bunch of different polyphenols and compounds from natural foods that are in and having an effect here, but it doesn't say which glucose transporter, how effective are they, et cetera, right? So it makes it a little bit difficult. Again, there's I have tons of graphics like this, okay? This is probably not even an exhaustive list of the graphics that I'm able to find in the literature, which are amazing, and it really helps me start to check these off the box, right? So what I was able to do is I took all those graphics in addition to other graphics, and I basically made this natural GLUT inhibitors sheet. And I took the ones that were the most kind of they were in the most graphics, I guess you'd say, because there may be 30 things here. I don't have enough room on my slide for all of them, but I found the ones that I could see the most often in a variety of papers, and then I cross-referenced them with their effects on glucose transporters. I think I stopped at GLUT9 just for my own sanity's sake. So I basically would type in the compound, let's say genistein, and it would show me that there were publications of it having an effect on GLUT1, GLUT2, GLUT3, GLUT4, GLUT5, GLUT6, GLUT7, GLUT8, and GLUT9, right? Talk about a pan-GLUT inhibitor. Genistein is a pan-GLUT inhibitor. It blocks all, or has effects upon at least all 9 of the ones that I did searches for. Another one in the running would be pretty good, would be Apigen. Apigen's pretty good too. You see one downregulation or inhibition of GLUT1, 2, 3, 7, and then there's some kind of finicky things here. So like some of these things will actually upregulate GLUT4, which may or may not be a bad thing for us. I'll digress and I'll explain that a little bit later. But upregulating GLUT5, which I believe is a fructose inhibitor, a fructose transporter, and then GLUT9 could also be potentially problematic. Although I'll be very frank with you, I have not seen, 4 through 13, I have not seen as being like major targets of pharma for these cancers. It seems like GLUT1 and 2 and 3 are the most important. So I think that when kind of like looking at this sheet and as a gestalt, I would say if it has effects on GLUT1, 2, and 3, it's going to be a candidate for very high potential. So that would be like genistein and resveratrol and curcumin and EGCG and catagens from like other catagens apart from EGCG and T, quercetin, hesperetin. Let's see here. This fluoretin, pretty powerful. Apigenin. So those are the ones that have the most effect on the broadest categories. Now let me circle back to like talking about GLUT4. So there are going to be, let's just go back quickly to this slide here. So there are some cancers that utilize GLUT4, right? So breast, lung, gastric, and pancreatic. That doesn't mean it's only using GLUT4. It just means that that is a transporter that it's being used by some of these cancers. GLUT5, GLUT6, all the way to GLUT12, okay? But you can see that the heavy hitters are really GLUT1, 2, and 3. And again, because we're not going to have a strategy that is based primarily and only on blocking GLUT transporters, we're going to be on a ketogenic diet. We're going to be using other things that block machinery within the actual glycolysis system later in the pathway. I think that my point is that if things even upregulate GLUT4, for example, let's go back down here. So for example, you see that, let's talk about resveratrol. Resveratrol upregulates or enhances GLUT4. So does curcumin. So does EGCG. Actually, EGCG I saw studies that showed that it decreased it in cancer cells and then it upregulated it in other cells, like normal cells. And then also quercetin. Okay. So my point is by saying this is that may not be a bad thing. The reason is because one of the ways, or a major way that resveratrol, curcumin, EGCG, quercetin, apigenin. You also see it here in silibinin or milk thistle. The ways that it actually gets rid of glucose out of the bloodstream is that it upregulates GLUT4, and GLUT4 is mainly in muscle cells, it seems. So my point is that when you exercise, you're upregulating GLUT4. When you take some of these compounds, it upregulates GLUT4 to get rid of glucose. Probably cold has a very similar effect. I have to look again at the mechanism. But my point is that is actually a way to actually steal glucose away from the tumors, which are mostly utilizing GLUT1, 2, and 3. So if you see an up arrow at GLUT4, that may not be, and it probably is not, detrimental to our story. And I just wanted to give that nuance, okay? Because I don't think it's going to be as detrimental. But that is what I've got for you when it comes to GLUT inhibitors, both, I guess, pharmacologic and drug and natural. And, you may already be on, let's say you may be already like doing your own version of Press Pulse or your own version of, let's say, metabolic therapy, right? Kind of unsupervised because you can't find anybody locally that can help you with this, which I completely understand because very few people know about this kind of stuff. But my point is that let's say you're on EGCG and you're on curcumin and quercetin, like you're already having a pan GLUT effect, right? On GLUT1, 2, and 3 with quercetin, GLUT1, with EGCG, GLUT 1, 2, and 3 with curcumin, GLUT 1, 2, and 3 with resveratrol. Let's say you pick those 4 things. You're already having a big effect. Maybe you're on milk thistle. You got to enhance GLUT 1 effect, apigen. And, my point is that you probably are already covering this. And then remember, if you are taking mebendazole, you're also having effects on GLUT 1 as well. So you may not need to really fret, I guess, about further inhibition of GLUT transporters because you're already on compounds that may be already blocking those things. I do think that what I'm probably most excited about with this particular lecture, okay, from a pharmacologic standpoint, I'm most excited about Glutor. Like if you type in Glutor in Google, you'll find it at a chem store. You can find it. The question is, we have got a lot of work to do, what to know about safety profiles, about how to administer it. Can it be taken orally? If it does, what dose do we use? Like we have no idea. So therefore, I would not be messing around with that if I were you, because I think we have too much to learn there. That being said, what I'm also excited about is, I don't know, the power of like Genistin, like something that I don't personally use in most of my protocols, but I may reconsider it. Resveratrol, I do use a lot. I do use a lot of curcumin and quercetin is some of my favorites. I use, silibendin. I don't use a lot of apigen, but maybe because of this work that I've been able to produce here, maybe that would be something that I would be considering, putting in to someone's protocol potentially, if they're having a really hard time getting their glucose ketone index down. Maybe some extra effort could be put in by utilizing some of these glucose blockers. And that may be a strategy, for some clinicians going forward. I don't think anybody really truly knows the answer yet, but this should move us forward in terms of having some more tools in our tool bag, both pharmacologic and non-pharmacologic. And I will say that I personally don't use the SGLT2 inhibitors for people, but if it's a tool that is useful and it's for a short period of time, because I'm just not a big fan of drugs, despite being a conventional doctor, make sense. Then I would say that then that could be something that we could do for a short period of time. The way I kind of look at metformin, that's the same way I kind of look at statins, in the context of cancer as well. Somebody just asked a question here. Is it just mebendazole or fenbendazole? So this is the issue. Because fenben is not going to ever be studied in humans because it's an animal medication, we'll never for sure know the answer to that. That being said, I do believe that given that fenben and menben are in the same pharmacologic category, they likely have the same effects. I'll take that with a grain of salt because I think it's going to take study, but my gut tells me that it's probably going to have the same effects. The next person said, would it be nice if I knew what GLUT transporter I needed to block? I know. Trust me. I mean, I think that's where the future, I think, should go. I think the future should be, instead of getting a biopsy to figure out what kind of, like, I don't know, what do you want to call it? What kind of genetic abnormalities your cancer has? Like, okay, who cares? We're going to look at what kind of GLUT transporters does your cancer have? What kind of glutamine transporters does your cancer have? Like, et cetera. So we know exactly what GLUT transporters to block and what glutamine transporters to block. So that way we can like maximally affect your cancer. That's the way I think it should go in the future. We're not there yet, obviously. Best safe to use combination then. Are you talking about a combination of fenben and menben? Or just a combination of multiple inhibitors? Your question is, should I combine fenben and menben? First of all, I can't give medical advice and I wouldn't tell you, but I don't think there's any reason to do both. I always err on using menben just because it's studied in humans. We know it gets past the blood-brain barrier. We know what the doses are. My gut tells me that fenben is probably sadly underdosed, just because I've seen other kind of alternative practitioners use menben at doses that are like jokingly low levels compared to what is used in studies. Menbendazole is a mg per kg dosing, and it's very safe, even up to 200 milligrams per kilogram. So true. I had a complete genome assay done and it didn't help at all. And I'm still being given 30-year-old chemo drugs. Yeah, man. Yeah. I mean, the circulating cancer stem cell test that Hem-Onc is using now. And I'm seeing, when I look at progress notes from my Hem-Onc colleagues here where I work at, it seems like these liquid biopsies, the Singletera's, et cetera, are being used clinically. And they're used regularly by Hem-Onc, conventional Hem-Onc. But I just, the way that they use it, or even the way that I've been asked to use it by, because some of these liquid biopsies will have like, it's sensitive to, because it has these genetic abnormalities, it's more sensitive to curcumin or whatever. It's like, look, I'm not doing it for genetic abnormalities. I'm doing it because I'm trying to block glucose, I'm trying to block glutamine, and I'm trying to hit it with oxidative stress as proposed by Seafried, as proposed by metabolic therapy. So I don't use it for that. I think the circulating cancer stem cells things are cool because you're able to see like, do you have active disease or not? But I think that the way that they're used clinically for oncology, I think once metabolic therapies take over, I think that they'll be superfluous and probably not helpful. All right. Well, we've went an hour and 1 minute exactly in 3, 2, 1, an hour and 1 minute. So I think we've pretty much hit this to the maximum amount that we can for now. If you guys like videos like this, please like it. If you have anybody who could utilize this information and benefit from it, please share it with them. And if you're not a subscriber to the Mitochondriac Army, please consider subscribing. It's free. And I'm going to answer one last question. Is there any success for Hodgkin's? What I'll say is that it's likely going to have effects across the board because these are hallmarks of cancer, right? It's just that it seems that the ones that we've studied today are mostly looked at for solid tumors. So, not hematologic malignancies. But that being said, my gut tells me that it's going to have an effect there. All right. Well, until next time, take care.